In this lesson, we'll introduce the concept of neuroplasticity, and we will learn that it is changing synapse strength that is the basis of learning and memory. First, some definitions. Memory is the capacity of an organism to acquire, store, and recover information from a learning experience. Plasticity is the ability of neurons to change the strength of connections with other neurons by growing stronger or new synapses, where the flip side is by weakening synapses or eliminating synapses, and this process happens during development. Now we're going to introduce an icon to represent a synapse that's getting stronger. Let's take a look at these two neurons here. There's a certain strength of connection between them, so the terminal is drawn with a certain size, and there are a certain number of neurotransmitters in the synapse there. Let's say this is baseline strength, and this is before a learning episode. If these two neurons are going to take part in a learning episode, we're going to represent the change here by showing the terminal. We're going to draw it bigger and have more neurotransmitters in the synapse, and that is intended to represent that there's a stronger connection, stronger communication between these neurons. The synapse is stronger, and this is a biological growth process. So this is what scientists think is going on when we're learning something, and if those stronger synapses can be sustained over long periods of time, that's the basis of memory. A memory trace then refers to the biological change in the brain that represents the learned information, and most often that will be the new or stronger synapses between neurons involved in the learning. So in our simple nervous system that we have seen before, memory traces are most often located in the central nervous system. In fact, we talk about the cerebral cortex being sort of the long-term storage site for many of our memories. But we're going to have our sensory system delivering information to the brain, and then the brain will integrate all that information along with our prior knowledge and experiences and so on, and then generate an output, some kind of behavior. So let's consider what kinds of ways that neurons can be organized in the central nervous system. Well, here we have three patterns. The first one, top left here, neurons can be organized into just chains, and this would be like a neural pathway. So neuron number 1 communicates with neuron number 2, neuron 2 activates neuron 3, etc. So we have a neural pathway. Over here, we have one neuron here. Neuron number 1 sends information to two different neurons in two different parts of the brain. Now, how can that happen? Because axons can branch. So the output signals going down the axon will go to both sets of terminals. So one part of the brain can talk to two separate parts of the brain. Information can diverge. Down here, we see the opposite. We see two neurons sending information to a third target neuron, the same target neuron. So here we have the convergence of information. And we'll see in a moment, this architecture, this arrangement is going to help us understand a simple form of learning. I should say one more thing just about the neurons in brains. They can host many synapses. This neuron down here in this picture, there's the cell body, and look at all these branches. Those are the dendrites. That's a lot of space for lots of synapses from other neurons. A typical neuron might have hundreds or thousands of synapses spread around its dendrites. In fact, if you look at the dendrites, there are these little yellow bumps. Let's magnify that. That's what this structure is over here. The brown part would be the branch of the dendrite, and the stained yellow here are spines. This is the location of any particular synapse. So roughly speaking, the number of spines will be the number of synapses. Now, in the way they prepared this tissue, we don't see the axon terminals, but each of these spines would have a terminal that makes the other half of the synapse. Here's just another picture of the dense, dendrite forest in a mouse cortex, and we see the dendrites here and all the little spines on here. And the point is that a single brain cell, and we have billions of them in our cortex, a single brain cell can make lots of synaptic connections with other neurons. So it can get very complicated up there in the cortex, but we're going to simplify the story to understand a simple form of learning. And we're going to use classical conditioning as our example. You may have heard of this process. So let's start it off with panel number 1 here. We have our friend here, our pet dog, and we present some food. Now the dog is going to see the food and also perhaps smell the food. Now you might know that that can automatically, in a sense, trigger salivation. Now we're going to call the food the unconditioned stimulus, US, and the salivation the unconditioned response. We didn't have to train the animal to do this. The dog's brain is kind of naturally wired up to salivate when in the presence of food. Look at number 2, though. Here we're going to come along at another time. Food is nowhere near, and we're just going to ring a bell. Now the dog might perk up and be interested and whatnot, but it's not salivating. So the way the dog's brain is initially wired up, a bell does not trigger salivation. Food does. Now what we're going to try to do is make it so that the dog does salivate to the sound of the bell. To do that, though, we're going to have to train the dog to associate the bell with food. How do you do that? You do repeated pairing. So you ring the bell and you present the food. And then at a later time, you ring the bell and present the food. So the dog's brain is being exposed to 2 stimuli, right? So the bell, which previously did not trigger salivation, but also the food is being presented, which did trigger salivation. If you do this training enough, at some later time, you just ring the bell, the dog will salivate to the bell only. There's no food in this scene. So the bell is going to be the conditioned stimulus. It's the stimulus that we're going to try to condition the dog to respond to, and the salivation to the bell is called the conditioned response, CR. So the dog is learning to associate the bell with food, that the bell predicts the food. Now, how do we model this with neurons? So here we go, before training, and we've got one of those scenes where 2 neurons are converging on a 3rd neuron, like we saw before. And we're going to identify the blue neuron here. This is in the bell pathway. So we'll say that when the bell rings, this neuron gets activated and sends signals to the green target neuron. When the food is presented to the dog, this red neuron gets activated and signals are sent to the target neuron. We should notice also, in addition to the convergence here, which will be key to this ability of the dog to associate these stimuli, we should notice that the strength of the synapses initially are different. The food pathway has a strong synapse, the bell pathway has a weak synapse. Now, why is that? We want to think of this as just being the way the dog's brain is initially wired up. There are lots of weak synapses in our brains as well, but they are there capable of supporting a new learning, and then we'll see what that involves here in a moment. So let's just think of this initial wiring as due to DNA. The genetic code of the animal has a role in wiring up the initial brain. Okay, so let's now take a look at what happens when the dog is prior to training. So it's a novice dog. So we go ahead and present some food, and sure enough, that's a strong synapse, so we're going to activate the target cell, and whenever the target cell is activated, we get salivation. So the food causes salivation. The unconditioned stimulus causes the unconditioned response. What about the bell? Let's ring the bell. Electrical signals race down here. It's a weak synapse. So notice the target cell does not get activated. If this cell's not activated, no salivation. So the conditioned stimulus initially does not cause salivation because the pathway that is representing the conditioned stimulus has a weak synapse on that target cell. Well, let's go ahead and do the training. So we're going to pair the bell with the food, the bell with the food, bell with the food. Notice when you're pairing the bell with the food pathway is activating the target neuron while you're also simultaneously activating the bell pathway. And as we'll see in a moment, that's going to create a condition where we can get some biological growth of the weak synapse. Let's take a look down here. Now this is after training. Notice we're going to ring the bell only. Don't present the food, just the bell only. And sure enough, we get salivation to the bell. Why? Because the pathway there, the bell pathway synapse, it grew stronger as a result of the training. So this is what we mean by plasticity. That plasticity is the ability of neurons to strengthen their connections. In this case, the training. The pairing of bell and food, bell, food, bell, food, bell, food, it created conditions where this synapse grew stronger. And that means the dog will salivate to the bell only. back to the concept of a memory trace. The memory trace would be this change right here. Remember, it's just the biological change that represents the learned information. Well, there it is right there, the growth of the stronger synapse in the Bell pathway. Now, let's take a look at two important conditions for this plasticity to occur. First, let's go back to before training, and we're going to have just kind of ring the bell. We're not presenting food. So this was like, let's see, this would be panel 2. We're just ringing the bell. Just ringing the bell. Okay, let's see. If you ring the bell, electrical signals will head down here to the weak synapse, but notice because this synapse is weak, the target neuron doesn't respond, so we get no salivation. No matter how many times we ring the bell, the dog never salivates to it because the conditions are not present to strengthen that synapse right there. And what's missing? Activity of the target cell. So the target cell must be activated by some combinations of its synapses if you're going to strengthen one of the synapses there. Got to have target cell activity. Let's look at another condition for synapse growth. Here, we're not going to ring the bell. We're just going to present the food. Well, if we just present the food, sure, we're activating the target neuron and we get salivation, but the synapse in the Bell pathway does not get stronger, right? Now, why is that? Because you need activity in this pathway for that synapse to be strengthened. So the synapse must be active when the target cell is firing if you want growth at that synapse. This kind of makes sense. Like if you just presented the animal with food and all of these other synapses got strengthened automatically, well then the dog would just automatically associate any a bell with food. If you just presented it with food, then it would just automatically salivate to the bell. That's not how the brains work. It's not how the conditions for synapse growth are specific here, so that you have to have activity in this pathway while the target cell is active. Then you can strengthen that synapse right there. So in summary then, a synapse grows stronger when it takes part in firing the target cell. And one way to think about this is neurons that fire together wire together. Now, you'll recall we said the initial convergence here was just the DNA instructions in that animal, and the fact that here, this synapse is weak, that one's strong, also related to the DNA instructions. But if the neural pathways for the two stimuli did not even converge, then the animal would not be able to associate those stimuli. So that would be a situation like this up here. If this were the Bell pathway and this down here were the food pathway, but they never converged on a common target, you wouldn't be able to associate a bell with food, right? So it's a certain architecture, and it's this architecture down here that supports associative learning. The Bell pathway and the food pathway have to converge so that you can create the conditions for neuron plasticity. You can strengthen a certain set of synapses, and that represents an association between the two stimuli. And of course, animals with larger brains have more areas in which information from the senses can converge in different ways, and this allows them to learn associations between various combinations of sensory stimuli. I mean, just think of language, for example. We have all kinds of words. What is a word? Well, it's just a sound that we make, but we want to associate that sound with maybe some object in the world, like the sound cat. We want it to be associated with the image of a cat, when we see a cat. Well, that's what brains are doing, associating things to things in our world. In this case, the sound, cat, and then the object, the furry thing that runs around outside. So larger brains then can support more memory traces because we have more real estate with which to kind of alter and modify synapse strengths and to associate stimuli in our world.